thank you everybody for joining us today. We have a wonderful webinar lined up. The eDNA Methods seminar series continues. Today we have a great lineup. We're going to be discussing and learning all about eDNA for harmful algal blooms, both freshwater and marine. The webinar is being recorded. You're able to watch this webinar afterwards. We'll have it posted on our YouTube channel. Links for that will be sent. We also have links always in our California Water Quality Monitoring Collaboration Network webpage. Today's webinar is being brought to you by two of the working groups under the California Water Quality Monitoring Council, the Molecular Methods Workgroup and the Collaboration Network, the one that I facilitate. If you have questions, when you're prompted to, you can unmute and ask your questions. You can always also submit your questions via chat. I'm really happy that we have Susie Throw from SQUIRP helping put this webinar series together. So I'm going to turn everything over to Susie and she can make introductions and tell you a little bit more about today's webinar and the really good webinars that we have coming up later this summer as well. Thank you. Susie?
Awesome. Thanks so much, Eric. And I'm joined today by Dr. Jamie Smith, also from SQUIRP, who will be taking you through a lot of the work that we're doing at SQUIRP with a number of our partners who I know are on the line and across California and beyond, working on developing molecular methods or DNA-based tools for harmful algal bloom monitoring. And as Eric mentioned, this webinar is part of a webinar series that we have through the Collaboration Network and the Molecular Methods Workgroup. If you've missed any of these, the recordings are all online. You can follow this link. And with that, I'm going to get into a little bit of introductory material to kind of just set the stage, a little bit of a refresher on what eDNA is, and then I'm going to hand it over to Jamie to take us through a lot of the work that she's been doing. Next slide, please. Okay, great. So just to start at square one, in case you missed any of our previous webinars, I think it's important to begin at the very beginning and define really what it is that we talk about when we talk about environmental DNA. So in its most basic sense, environmental DNA is the term that we use to refer to all the genetic material that's shed by organisms into their environment. And that can be into water, into air, into soil, anywhere into their environment. All of that eDNA is entering the environment often through the shedding of skin cells. It could be mucus, it could be saliva. All of that eDNA material is being introduced into their surrounding area where then we can go along in our boat and sample it out of the water column or sample it out of some benthic mats. And then we take that sample back to the lab, and as this workflow shows you here, we'll often concentrate that DNA down, extract it, remove any other particles or inhibitors that are present in the sample, and then we'll analyze it using either DNA metabarcode sequencing or quantitative PCR and a couple different methods that I'll get to in the next couple of slides. We really find a lot of advantages in using these eDNA-based approaches because they're really sensitive, they can be really accurate, they can be affordable, and they help us scale a lot of the questions that we're looking to answer, which we'll hear about more from Jamie later on. Okay, next slide. Given a number of the key advantages that these eDNA-based tools allow us to access, there's been this rapid increase in the number of different publications that are applying molecular-based methods, either DNA metabarcode sequencing or quantitative PCR, to investigations on harmful algal blooms, and in particular, how it's related to eutrophication or nutrient over-enrichment. And you can see in this figure here that over the past couple of years, we're seeing on the order of a couple hundred publications that are coming out annually where people are applying these DNA-based tools for better understanding harmful algal bloom events and the ecology of these organisms and some of the triggers of their blooming events. Next slide, please. I think did we skip one? Or is that the next? There we are. Okay, great, thanks. So there are a couple of different tools in our toolkit or our toolbox when we're looking at using these molecular methods for better understanding harmful algal blooms. And as I said, both their triggers and kind of the ecology of these organisms themselves. And one of the ones that we see as the most popular is using quantitative PCR, or its relative digital PCR. And quantitative PCR, hopefully a lot of us are familiar with this thanks to the pandemic and all of the qPCR tests that we've gotten, but it's really excellent in answering questions related to the abundance of key gene targets that are present in the system or of key organisms present in the system. So quantitative PCR is the tool that you would want to apply if you have a really species-specific or gene-specific question and you're wanting to quantify, say, the number of microcystis targets that you have present in your sample. Quantitative PCR would be a really great way of doing that. If you had an organism that was present in your sample and you didn't know who it was, you sampled a benthic algal mat and you want to know which species of cyanobacteria it is, then you would apply something that we call DNA barcode sequencing. And this is using an approach where you're taking a snippet of the genome, what we call a barcode, and we're using it to identify who the organism is. And you do that by comparing the DNA sequence that you generate to a library of all the previously published DNA reference sequences. If instead you have a sample, maybe you filtered some lake water and you want to better understand the community composition of all of the different cyanobacteria or all of the different dinoflagellates that are present in your sample, then you would use an approach that we call DNA metabarcode sequencing. You're using that same basic principle where you're taking just a snippet of that DNA and you're sequencing it to identify the presence of the organism and determine who they are taxonomically. But instead of just looking at one individual, now you're looking at the whole suite of the organisms that are present in that sample that you extracted. So it's a really great way for getting a more holistic picture of the broad scale biodiversity of the organisms present in your sample. Next slide, please. Awesome. So as I mentioned, there's a lot of great research being done taking these eDNA tools and applying them for harmful algal bloom monitoring. And this paper in particular that just came out a couple years ago highlights some of the most popular ways that these tools are being applied in the harmful algal bloom space. So as this figure shows you here, really, we've seen the largest growth in the application of eDNA-based tools using quantitative PCR and digital PCR. As I had said, these are really great if you have some species-specific questions or if you're really interested in quantifying maybe the genes that are involved in the biosynthesis of toxins, or if you're interested in quantifying individual harmful algae that you know might be present in your system. I would argue similarly, DNA barcode sequencing has become really popular, as I said, for getting more of that holistic view of the harmful algal bloom community. And one of the tools that we see emerging now in the field are some of these isothermal assays. That means they can be done out in the field. They're really rapid. Some of them rely on a CRISPR or a LAMP assay, again, that we've seen developing also in the COVID testing world. So they can give you really rapid feedback and analysis along the lines of like a pregnancy test, where you can have a visual analysis that's done on the spot in the field to identify whether an organism or its toxin are present in the field. That's definitely something that's emerging kind of on the vanguard in the harmful algal bloom space, but I think that we'll see some really big development in that arena as well over the course of the next few years as people get increasingly interested in applying these eDNA-based tools for better surveying and having early warning indicators of harmful algal bloom species and their toxin production that might be occurring. So hopefully that gives you a good 5-minute overview of some of the tools and techniques that we've been using, that other folks that are interested in harmful algal bloom monitoring are using and applying and incorporating into a number of our different biomonitoring programs. And now I'm going to hand it over to Jamie to tell you specifically about some of the work that we've been doing here in partnership with a number of folks across California for developing these tools for harmful algal bloom monitoring. So with that, I'll hand it over to Jamie. Thanks, Jamie.
All right, great. Thank you, Susie. Can you hear me okay? Okay, perfect. All right. So Susie gave us a great overview of some of the techniques and that are kind of in place or are up and coming for harmful algal bloom monitoring. And so I just want to give you a little primer on harmful algal blooms themselves, and then we'll take you on a tour of kind of how these techniques have been applied over the last several years in California to better understand and monitor harmful algal blooms. So to start, harmful algal blooms are a really interesting research question and also a big monitoring challenge, and we'll get to that in a second, because they are diverse. So we have harmful algal blooms in marine systems. There are a number of different taxa that can produce harmful algal blooms that vary across regions. So in California, because this is a very California-centric talk here, Our main issues were with harmful algal blooms formed by diatoms. So you might have heard of Pseudo-nitzschia as our main kind of HAB-forming taxa coastally, as well as some dinoflagellates like Alexandrium, Lingulodinium, Dinophysis, as well. And so we have a number of toxins that are made by these different organisms. So again, domoic acid is probably the one that gets the most attention here in Southern California because it has caused, or in California as a whole, it's caused kind of the most issues, things like stranding California sea lions and whales and dolphins. But we also have issues with saxitoxin, okadaic acid, yoxitoxin, and other toxins as well. So in addition to kind of what we have going on in marine systems and our inland freshwater systems, we also have issues with primarily cyanobacterial blooms in our, lakes, rivers, streams, ponds. And cyanobacteria are very, very diverse, showing, again, some pictures of some of the different genre and species here. And they produce a number of different cyanotoxins, which is the toxins that cyanobacteria form. Probably the most common one that you've heard is microcystins, but there's also anatoxin A, cylindrospermopsin. Some actually produce saxitoxin as well as a variety of dermal and emerging toxins. And so we're dealing with a lot of diversity here in California when it comes to harmful algal blooms. And so there's some unique monitoring challenges that we face when dealing with harmful algal blooms. You kind of have two pieces of the puzzle, which is the detection of the toxigenic or toxin producing cells or species, and then the toxin production itself. And in many cases, or most cases, actually, the species that have the ability to produce toxin can regulate their toxin production. So they're not always producing toxin. So you kind of need the presence of the cells and an environmental trigger for the toxin production to occur. And so when it comes to kind of the difficulties with monitoring the toxigenic cells themselves, a lot of times there's multiple toxin producing species or strains, and we'll kind of show an example of that in a couple minutes. They are sometimes very difficult to ID, or you need a lot of practice identifying the different taxa because of the diversity that we talked about. I think we have somebody that needs to move. Awesome, cool. Sometimes we can see issues at very low abundances of these toxigenic cells. So sometimes they're really at levels that are hard to detect using traditional microscopy approaches. And like I said, or cell presence doesn't always equal toxin presence. On the toxin side, toxin can be ephemeral, and so just because you don't measure toxin today doesn't mean it wasn't there yesterday, isn't going to be there tomorrow. Some taxa can produce multiple types of toxins at a time. So you're dealing with multiple toxin targets. Effects can happen at very low levels, and toxins, again, because you're dealing sometimes with multiple, can be very expensive to measure. And so eDNA provides us with a lot of powerful tools within this kind of nexus to be able to monitor as well as understand the ecology of these events. So there's a lot of great specific questions that eDNA can answer, right? We can say, what species are causing a bloom? You walk up to a lake, like this is Clear Lake a couple years ago, and say, well, what is causing this bloom? eDNA can help you answer that. What are places that are at a higher risk for toxin producing blooms if you have a number of lakes that you're managing and you want to understand kind of what's going on in these different lakes from a HAB potential. You can say what are the potential causes of toxin production. You can look at the regulation of genes or the uptick of genes in relation to environmental conditions. You can say which of the specific taxa that seem to be producing toxins. There's a number of approaches using eDNA where you can get at that question, as well as how is your community composition changing over time. So sometimes, we'll get into that, but you can have shifts in your dominant taxa and shift from different types of toxin producing species. So I'm going to take you on a tour of a couple different applications of this. So let's start on our coastline and how we can use environmental DNA to learn about domoic acid producing events. So what I'm showing here is a nearly 15-year time series of domoic acid along the California coastline from a combination of monitoring and research efforts. And so I think what is striking about this figure is the up and down of the domoic acid concentrations, where some years you have very, very high levels, some years you have very, very low levels. And what's interesting to ask is, what are the drivers of this type of variation? So I'm singling out 2 years here where we did some work to try to understand why we had a really kind of high toxin and quote unquote low toxin year between 2013 and 2014. So using some traditional approaches, you can take a water sample and you can do microscopy and count the individual Pseudo-nitzschia cells. You can identify them typically to genus level. It's actually, you know, the species level determination via microscopy is pretty difficult / impossible. You require like SEM, and it's really difficult to do. And so with using traditional approaches, you can count kind of your cells per liter. And so what we have here is an instance of a Pseudo-nitzschia bloom in 2 different years, where we have very similar cellular abundances. But if you look on our y-axes, we have an order of magnitude or several orders of magnitude difference in toxin concentration. And so the question, that really arises here is, well, we have similar number of Pseudo-nitzschia present. Why are there such large differences in the toxin concentrations? And so we can turn to our eDNA tools to really kind of get at this question. And so here we used a fingerprinting technique called ERISA to look at some of the samples and look at the community composition of the different types of Pseudo-nitzschia species, because there's about 50 plus species and only about a third have the ability to make toxin. And so what I'm showing here in this figure is the difference between 2013 and some samples in 2014. And I've color-coded the species by what we know about their ability to produce toxin, right? So with the warmer colors being the ones that we know can produce a lot of toxin, kind of going up as you get to kind of yellow and orange, less prolific toxin producers, and the ones in black and white are ones that we don't think produce toxin. And so you can see pretty easily here using this fingerprinting technique that really one of the big drivers between our high toxin year in 2013 and our kind of moderate to low toxin year in 2014 has to do with the different species that are present within that Pseudo-nitzschia community, which if you just did cell counts, you couldn't really tease this type of ecology apart. So the diversity within the Pseudo-nitzschia, the interspecific diversity that is within the community, has really important ecological implications. So that's one example kind of in marine systems. And moving now into some of our inland systems, there's a lot of work that's been done to understand cyanobacteria blooms in our lakes throughout California. So we'll start with some work that's been done in Lake Elsinore that was published a couple years ago by Meredith Howard. And Lake Elsinore is a lake that does experience really chronic persistent blooms of cyanobacteria and very high levels of toxin. So here I'm showing you a measurement or a graph that's showing the presence of 3 different types of cyanotoxins. So microcystins in the black line with cylindrospermopsin in the blue, and then the dotted black line is anatoxin. And so basically throughout this summertime survey that was done, all 3 types of toxin were produced, but they're peaking at different times, right? And so we see kind of an initial peak in anatoxin, again, in that black kind of dotted line, then cylindrospermopsin in that blue, and then later we see a big giant peak of microcystins. So for reference, the California danger level for microcystins is at 20 micrograms per liter. So this is several orders of magnitude above that danger tier 3 human health trigger level for the most of the rest of the year. And so again, how do changes in the cyanotoxin level, if you were just monitoring the cyanotoxins, it would be, this is great for human health. We know, to put up postings based off of this, but we don't really have a lot to explain why this is happening. So we can ask the question with eDNA, how are the changes in cyanotoxin levels related to changes in our cyanobacterial community? And so over this same period, now I'm showing some metabarcoding results, and you can see that there is kind of a distinctive shift Don't necessarily even have to worry too much about, exactly what the different colors are, but you start with a planktothrix, and then you shift over to a microcystis dominated community. And so I kind of just pointing out here that during this time period that you have that shift in community composition, you see that corresponding shift and spike in those microcystin levels as well. So we can learn a little bit about, okay, this is why we start seeing the shift in toxins and that big spike in toxins as well, by using eDNA to really understand what the specific community is doing over a summertime season. And kind of moving up north to Clear Lake, we can kind of ask similar type questions, and you can use these techniques over a longer period of time. And in a place like Clear Lake, where you have annual kind of high density cyanobacteria blooms, I'm showing kind of pictures here of the different blooms over a 3-year period. You can ask, what's going on with these blooms? Are they the same every year? Are they different? And what can we learn about the drivers of these blooms by looking at the blooms over multiple years? And so we kind of, in this work, which is out of Dave Karen's lab, and his postdoc Isha was gracious enough to share her data that's in preparation with me to kind of go over this. But this is an interesting kind of set of blooms that was captured, where there was obviously a high biomass bloom in 2019, but very little toxin detected. In 2020, there was some toxin detected, but not at very extreme levels. And then in 2021, there were kind of higher level toxins that were up above our or near our California warning human health trigger. And so you kind of have this scenario of 3 different years, kind of 3 different manifestations of the presence of microcystin. And so we can then zoom in, again, using our eDNA techniques and using metabarcoding to take a look at what the community is doing in different years. And so here I'm showing you kind of what the communities look like at those different points in the lake during our high, medium, and low years. And you can kind of see, again, by looking at the colors, that there are distinctive communities that are occurring. And I'm just going to overlay here the asterisks are where toxin was detected within the lake in each of these years. And so you can see that there's some really cool interannual variability in community composition, as well as variability throughout the lake. This is a very large lake. And so you almost kind of get these areas that are very distinctive communities. And so what we see is that Microcystis dominated in the lower arm site in 2019 and 2021, whereas Limbia in that middle year in 2020 was our most dominant taxa. And then in 2020 and 2021, Delicuspermum was dominant in kind of the upper arm and Soto Bay and Narrows area, whereas that same kind of region was dominated by Cyanobacteria. And so we can kind of start to understand what are the species that are associated with the presence of toxin using this type of approach. We can also kind of use eDNA to say, well, that metabarcoding data was focused in on cyanobacteria, but you can do some additional sequencing and look at the other kind of microbes or algae that are present in the samples, and start to kind of ask questions about, are there potential relationships with other eukaryotes like algae and fungi? And so what's interesting is that we do see kind of along with our distinctive communities of cyanobacteria, here we also are seeing kind of distinctive eukaryote communities as well, where in 2019, most of the samples were dominated by chlorophytes. In 2020, we see diatom dominance. And in 2021, we see a combination of chlorophytes and cryptophytes dominating. And so we can start to understand how things like potential food web interactions or interactions with other algal groups might relate to the presence of toxin. And so that's some work that Isha is really working on, kind of teasing that apart right now. So this is just a taste of that work. So I just kind of showed you a couple instances where we've used kind of metabarcoding across time to try to understand dynamics in cyanotoxins. eDNA is also a great tool for kind of doing kind of a risk assessment type survey. If you have a region, say, where we don't know a lot about what your risks might be for HABs, you can use a variety of approaches to try to understand, you know, if you have a limited number of monitoring dollars, where do you really want to spend your time monitoring versus places that might be kind of a quote unquote lower risk, and you do that in a data informed way. So we did that in 2020 through the LA region, where we did a pre and post Labor Day survey of 17 recreational lakes, where we sampled for a number of indicators, including cyanobacteria community composition, like we showed you in the last couple lakes, looking at microcystin producing genes, looking at cyanotoxin levels, as well as kind of some corresponding environmental correlates like chlorophyll and nutrients. And so by using kind of those indicators together, you can get a picture of what might be going on with your lakes. And so we used a combination of grab and passive samplers to look at microcystins. And so looking across those lakes between the two sampling events, 53% of them had some detectable microcystins. And then when you use your eDNA tools, you get kind of an additional picture of, okay, well, when you look at your microcystin toxin producing genes, 88% of the lakes had the genes that could produce, I'm sorry, I switched that. When you look at your barcoding, 88% had taxa that could be microcystin producing taxa. And then when you look at the genes, 100% of the lakes had the presence of the microcystin producing gene. So that second piece might be a little confusing, but as we don't necessarily know all of the taxa that can produce microcystins, so we kind of use environmental and research studies to understand that. So what we're kind of seeing here is that with our current knowledge of the taxonomy, 88% of the lakes have microcystin producing taxa, but we detected the gene in 100% of those lakes. And so that can kind of start to give you a picture of, okay, well, all of these lakes have the genetic potential through looking at the gene potentially to make the toxin. 53% of them actually had the toxin present. And you can sort of start to understand where your higher risk lakes are when you move from kind of the idea of potential to actual presence, to help kind of understand where you maybe want to focus your monitoring efforts. And so by looking across a region in this way and using these different indicators, you can also learn about which kind of toxin producers. So that 88%, when you drill down, here's just a subset of some of the lakes that had microcystins present. You can kind of go and drill down and say, well, what are my potential toxin producers? Again, if we're going to use it like, a combination of molecular and microscopy, you can start to kind of come up with a hit list of your characters in your region that you want to worry about, or within specific lakes that seem to be most associated with the presence of toxin, and use that to kind of start to design your monitoring programs for some of these lakes. So here, you're looking at 4 of these lakes within that region. There's at least 5 different potential microcystin producing cyanobacteria genera across these different water bodies. And then you can also use your gene probes to identify your potential toxin producers. So here I'm kind of showing on the y-axis the digital PCR results. So you get kind of a copy number of the presence of those genes, which again is kind of your genetic potential to produce the toxin. And then on the x-axis, we have the actual microcystin concentrations themselves. So you can kind of see as you kind of go up in your toxin counts, or your gene toxin counts, as you increase your toxin concentration, that kind of correlates. It's not perfect, right? Because this isn't necessarily going to work super well if you go across multiple systems, but you can kind of ask the question of, like, we used a gene target that was Microcystis specific. And so we can kind of say that, okay, microcystins are often, but not always, going to be produced by Microcystis across these lakes that we surveyed. But you can use your gene targets. Sorry, I'm not going forward. Your gene targets are really useful in the context of monitoring in an individual water body, where you can start to apply those gene targets in a more quantitative sense of helping you understand when you maybe want to start measuring for toxins. So here I'm showing a time series where one of the lakes we returned to in 2022, and we're monitoring every 2 weeks over the summertime period. And so here we're showing in the orange, I'm showing kind of the microcystin levels that went up over time at Lake Machado. And then we were also measuring our microcystin gene targets that were Microcystis specific over that same time. And you can kind of see how you have that potential, and then your counts kind of go up at the same time that you start to see the actual toxin being produced. And so toxin genes are really powerful in that they're actually sometimes cheaper than measuring the actual microcystin or cyanotoxins. And so they can help kind of inform, if you're kind of trying to make a tiered monitoring program, you can use your gene target as kind of a first line of monitoring that's a little bit more cost effective to help you identify which samples you maybe want to test specifically for the actual toxins as a cost saving measure, but while also being able to keep an eye on what's going on over a time series, right? And so what that could look like is, this is kind of what the HABs program within SWAMP is kind of envisioning, is a lot of places can employ a tiered approach to assess freshwater blooms and cyanotoxins, where you can employ a number of techniques, starting with kind of just visual eyes on the water body, collecting samples, and then determining using either your microscopic or barcoding approaches, looking at the taxonomy or looking at your gene counts, what toxins you want to measure and when you maybe want to start measuring, so that you can target your analytical dollars towards the right toxins that are going to be your biggest concern at the time that you see that toxin gene count or your toxigenic species coming up, in order to kind of keep routine eyes, but also, again, kind of balance the costs of the analytical analysis of the cyanotoxins themselves. So that's kind of a great approach using our qPCR, dPCR that Susie mentioned at the beginning of the webinar, within a monitoring framework. And so within, that was kind of, again, within inland water. So kind of coming back to our coastal waters, we haven't had that capability in coastal waters because of most of the genes pathways associated with our coastal blooms were not elucidated. And so we were kind of left with measuring Pseudo-nitzschia and measuring domoic acid, and trying to use those two to help understand and monitor for blooms. But recently, the gene pathway was elucidated for domoic acid as well. And so kind of one of the things that we can hopefully employ in the future is using that same kind of genetic approach to look at your genes as hopefully an early warning of toxic events, as well as understanding when toxin genes are kind of coming up and down in relation to when you see the toxin, in relation to what environmental factors are present, to better understand the drivers of toxin production for domoic acid producing blooms. So I took you on a tour of a couple different scenarios of applying eDNA within a monitoring and research context. And so kind of just to wrap up, some final thoughts and conclusions is that eDNA approaches are really useful and can sidestep some of our issues that we have with morphological IDs. So for example, bringing it back to that first example where you can't tell your Pseudo-nitzschia species apart using your traditional morphological IDs using a light microscope, you can use eDNA approaches to do that and better understand your ecology, or when you are seeing the very concerning species in the region. Sampling across various different lakes has shown us that there's a lot of cyanobacterial diversity across California, as a state, as well as within different regions, as well as diversity of what your toxin producing genre might be. It's not just Microcystis that causes issues. And paired observations with taxonomy and barcoding with measurement of toxins helps to better understand what our problem taxa might be across a region, within a specific lake, or across the state. And so eDNA can really help us understand our cyanobacterial communities better across the state in order to better address the management, because different types of species have different kind of ecological niches. And by understanding, who's causing problems where, you can apply a customized management approach to address the potential issues with those different taxa that are causing issues. And lastly, kind of moving towards an understanding of drivers, we can kind of pair, eDNA is a really powerful tool to pair with the other kind of tools in our monitoring toolbox, such as characterizing our toxins and our environmental conditions, and building a better understanding of what are kind of the triggers and causes of the biomass accumulation, as well as the toxin production across our various lakes. So with that, I think, Susie, did you want to go over this last slide, or do you want to just do questions first? Okay, let's go ahead and with that, I'd love to kind of take any questions, and then Susie at the very end will have some kind of housekeeping items to go over. So thanks for your attention, and happy to take questions.
I'm trying. You know what? I see one question in the chat. Jamie, do you want me to read it?
Yeah, can you help me moderate? Yeah.
Yes, yeah. So in the chat, there's a question. How can eDNA monitoring be applied to treatment, timing, and methods?
That's a really great question. So I think, as I mentioned in that kind of concluding slide, understanding which taxa are causing your issues helps you have a more targeted approach, because there's taxa that have the ability to fix nitrogen, that are able to regulate their buoyancy. And so there's little ecological niches of these different cyanobacterial genera. And so by applying eDNA to understand who's causing your problems, you can select an approach that is most kind of tailored to address the toxin production by those specific taxa.
Awesome. Thanks, Jamie. I will keep going. We have a question from Vivian that says, was allopathic power considered when looking at multi-species succession in different water bodies?
Mm-hmm. Do you mean allelopathic?
Maybe.
Okay, yeah. So I think that's a really great question, and putting my HAB ecologist hat on, that's really a fun kind of research question to address, because I think that these taxa definitely interact with each other in a variety of ways. The nature of the interaction, I think, is really unclear, and we're just starting to understand how these interactions work, because I think you can have kind of antagonistic allelopathic type interactions, but you can also have kind of mutualistic interactions as well that kind of help promote your toxin producing species. So in the example that I showed in Clear Lake, I think that's part of the goal with looking at the entire kind of microbial and eukaryotic community, is to better understand, are there associates that either kind of lead up to the production of toxin or the demise of a specific toxin producing taxa. So again, that's a really fun, interesting research question that we're just starting to understand, but eDNA is super powerful to get at that, because we can get kind of our most holistic understanding of the community.
Awesome. Great. Next question from Christina. Is there a public tool that keeps up-to-date records of saltwater HABs in California?
A public tool that keeps up-to-date records of saltwater HABs. I think our best tool would be from the HABmap program, which I can put a link in.
Drop a link.
Yeah, and so that currently is not employing eDNA approaches, but they do microscopy and weekly monitoring of domoic acid, and that data gets uploaded onto a NOAA-hosted server from, I think, at this point, 9 different locations across the California coast, and that's probably our best long-term record of marine HABs in the state.
Awesome. Great. Next question from Zach. Great talk, Jamie. Are there available digital PCR or quantitative PCR tools for monitoring domoic acid genes?
Mm-hmm. So I think those are really, that pathway is relatively recently discovered. The paper came out in 2018. And so I know that came out of the Andy Allen's group at Scripps. And so they're working on kind of making that into a commercial type assay in the way that it is available pretty easily on the inland side. But I think it, like, as far as monitoring applications, they're still kind of working out which genes in the pathway are the best ones to target. And so that is probably in the next year or 2 years gonna be coming out as a tool that works in kind of marine systems. So it's kind of an up and coming thing. It's in process.
Awesome. I think related question, I'll jump over to this one. Is the qPCR assay for the microcystin gene targeting eDNA or eRNA?
Good question. So the way we applied it, it was eDNA. So we just did a genomic DNA extraction. And so that's why, often you'll see where you see the genes, but you're not necessarily seeing the toxin. We're not looking at the transcripts. There are ways to look at the transcripts. It's definitely a lot more intensive because your RNA is going to change, on the order of less than an hour, and so the handling of the samples is a lot more intensive, but it is something that can be done as well.
Awesome. This is a good one from Michael. We have, could there be an instance when bloom senescence results in no eDNA but persisting cyanotoxins?
Absolutely, yeah. We do see that where, especially in our land to sea interface zones, we often detect things like microcystin in our coastal zones, but we're not really necessarily picking up microcystis or other toxin producers in the region. And so you can have instances where the toxin gets transported and the producing cells are not present. So, yeah.
Awesome. Okay, I'm going to keep going because we have enough time. This is like rapid fire Q&A, so I like all these questions coming in. Okay, how much in percentages and under what conditions do different cyanobacterial species actually produce toxins?
Oh.
So how much in percentages and under what conditions? So I would interpret that question like, what percentage of cyanobacteria species produce toxin and under what conditions?
Yeah, that's a really great question. So I'll take the second part first of, I think we know that there's things like changes in temperature or nutrient stress. Allelopathy came up, so we think that interactions with grazers or other kind of antagonistic interactions with the community can lead to the production of toxin. We don't the hard part in understanding what the triggers are is we don't fully know why the cells are producing toxin and what benefit ecologically it gives to them, but those are kind of, again, especially in marine systems, but in freshwater as well, the nutrient stress and the interactions is one of some of the leading hypotheses of what kind of triggers the toxin production. And then what percentage of cyanobacteria have the ability to produce toxin? I think I would struggle to give you a good answer on that because I think the cyanobacteria are so diverse. It's hard to really give a good solid estimate because not all of them have been brought into culture and tested for their ability to make toxin. But they are ancient, and I'd say probably a lot of them can produce a lot of different secondary metabolites that happen to be toxic to us or harmful to humans and animals.
Awesome. Okay, this is a great one. I think this is something you and I are both really interested in. I think this is from Kale. Is there a consistent method and procedure for eDNA cyano monitoring in California?
That's a really great question. I'm happy to tag team that with you, Susie.
Sure,
Is that,
yeah.
not really, but it would be great if there was, right? And then
Mhm,
I think
mhm.
that's, maybe you can speak to kind of what the molecular work group is trying to do and on that front as far as standardizing approaches.
Great. Perfect. Yeah, actually, you want to put that slide up.
Yeah.
I would 100% agree with Jamie. We would love to see some greater standardization or harmonization in the ways that we're sampling cyanobacterial communities and then processing them and analyzing them. And it's something that the Molecular Methods Work Group is working on in collaboration with HABs, Harmful Algal Bloom Network. And a lot of the methods that we're applying are really similar. And so it would be great to be leveraging some common resources, so the reference databases that we're using, making sure that they're consistent across different monitoring programs and research groups. And so that is something that the Molecular Methods Work Group has been directly focused on. And so if you are new to the Molecular Methods Work Group, it's a work group that we formed a couple of years ago to do exactly this, which is enhance coordination in eDNA-based monitoring and develop standardized operating procedures and best practices for environmental DNA sampling and analysis. And so it is a diverse group of folks, all from eDNA novice to expert users across a number of different research programs in agencies and stakeholder groups. And so if this is something you'd be interested in doing or participating in, we meet monthly. It's open to everyone. And it's really just about developing a community and the resources to support DNA-based monitoring across California. So that was a perfect plant for this slide and to tee off the work of the Molecular Methods Work Group. So thank you for that question, Kale. And with that, just a time check. It looks like we have a little bit of time for a couple more questions. Jamie, do you have stamina for a couple more?
Yep.
Okay, perfect.
Keep it going.
Okay. How do you account for the massive portion of metabolically relevant and transposable genes in the genomes of cyanobacteria when generating taxonomic assignments and assessing toxin production potential? As Vivian brought up, changes in cyanobacteria protein cycling and function are largely controlled allelopathically versus as a result of transcriptional oscillations.
Wow.
Yeah, this is an advanced level question.
This is I think that so one, when we're doing barcoding, we are trying to choose a conserved region. So we choose 16. A lot of times we work on the 16S, or we do like a cyanobacterial specific kind of 16S primer. So from a just identification standpoint, we try to choose places that are not changing as rapidly. So I think that's one way that we try to address that. From a metabolic perspective, I think that's really challenging to address, and I think that let's see. So in relation to the toxin genes in particular, um, we do try to verify that. I think that's why you don't want to ever rely on one kind of like toxin genes alone because they're going to act differently in different taxa. We showed you that we used primers that were kind of Microcystis specific for some of our work. And so I think that's one way. But I think that the thing is that you can never get these are all just different ways to try to understand how these things are working, and we're trying to get in the ballpark of our true answer, and we're never gonna like have like the most perfect method that can address like every single biological issue, because biology is messy. So we just are kind of doing the best that we can. I don't know, Susie, do you have any
Yeah, no, I
anything to
think
add to
Jamie, you
that?
hit on it. Like the one part of that question is exactly what you said. A lot of times for identifying taxonomically who the taxa are, we're using gene regions that we target conserved areas that flank these hyperdiverse regions. They allow us to better understand phylogenies of these organisms, but that's what we're often using, something like a 16S for doing our identification of who the cyanobacteria species is. When it comes to determining if we think an organism is producing a toxin or not, I would say by and large, we're still doing that through a culture-based approach. But sequencing or targeting or quantifying the genes that we're finding in the system are giving us an idea of, as Jamie said, the potential. And it's really great to be able to pair that with other lines of evidence to get a better sense of who the toxin producers might be in a system. So yeah, I would agree with you there, Jamie. Awesome. Hopefully that answered your question, Stuart. And okay, great. Just a couple more here. So we have a question from Max. Thank you for the informative presentation. In the case of microcystin, can eDNA help in identifying the types of cyanobacteria besides microcysts producing the toxin, and how?
Yes, absolutely. So I think there's a number of approaches. So we presented qPCR. That kind of infers that you are targeting, something relatively narrow, and you have an understanding of who your toxin producers might be. And so there's different specific kind of primers depending on, we used a microcystis specific target, for example. There's ones that are more genomically broad or taxonomically broad rather, or focus on different species. But if you really don't have a good sense, there are more advanced eDNA type approaches like metagenomics, where you can start to look at your genes present and try to attribute them to assemble essentially gene metagenomes and try to attribute your toxin producing gene to a specific taxa. And that's bioinformatically a lot more intensive, and I'm definitely not like the world's expert on that. But there are ways, if you're like, oh, I don't know, I'm seeing I'm not seeing any of like the normal taxa. You can kind of assemble metagenomes, for example, and try to see where that gene kind of fits taxonomically.
Awesome. Perfect. Okay, second and last question. How long does DNA typically persist in the environment, and can eDNA sampling methods determine how long ago DNA was shed by an animal? I can take a stab at that one if you want, Jamie.
Yeah,
so great question. I would say this definitely falls right in the frequently asked questions of eDNA. And I would also say it's one of the ones that we don't have an easy answer for. It's an area of active research. And it has a lot to do, so the decay rates of eDNA will be influenced by different environments. So water temperature, nutrients, pH, UV exposure, nutrients in the system can have an impact on eDNA decay rates. And so oftentimes we're trying to better understand the environment to better predict these eDNA decay rates. I will say studies so far have shown that it persists on the order of a couple of hours to a couple of days. Is the average that we see in typical marine and freshwater systems. And so can eDNA sampling methods determine how long ago eDNA was shed? And this is definitely an area of active research, and folks are incorporating a number of statistical approaches to try and get at that, and using experiments where maybe you know the source, where eDNA is being shed or deposited, and then you are able to kind of back calculate how long ago it was deposited or shed based on how concentrated it is in the water column. Another way to get at a question regarding timing of when eDNA is present in the system is actually, as was brought up before, using eRNA. So we know that eRNA has a much shorter lifespan in the environment, and so you can get more refined estimates of activity or presence if we're targeting eRNA. But that's an area of active research, I would say. So hopefully that answers your question.
And can I add something to that?
Yes.
Is in the cases of what we're dealing with in HABs, a lot of the time you're just collecting the actual cell because these are microbes. So a lot of times you're just filtering your water and you're collecting your actual cell. And there's a step in the extraction where we're actually like opening up the cells to get the DNA out. So it's not always, in the case of this application, we're not always dealing with like shed DNA, but you can have a combination of your actual cells and then exogenous DNA that's bound to like sediment or something as well. But yeah.
Awesome. Yeah, good point. Great. Okay, last one. With eDNA monitoring, are outside factors of water temp, air temp, et cetera, being recorded related to growth, toxin production, et cetera?
They should be.
Yeah.
They're not. Yeah, I'm a big
Right.
proponent of, yeah, I understand that like you can apply different tools in different ways. And if you want to understand, your human health threats, you have one kind of application of these tools. If you want to understand the drivers of what's happening, you do need to kind of get those environmental correlates. And so understanding your water temperature, your precipitation regime, all of those different kind of ancillary measurements can really help to untangle, in the, like, I think of the Lake Elsinore example where you had a shift from one dominant taxa to another. You're like, well, what maybe happened if you measure nutrients and temperature and water column stratification, you can start to kind of understand some of the conditions that might have given rise to that shift in dominant taxa. So yeah, that definitely, if it's not, that I'd highly recommend doing those things if you can in your programs.
Awesome. Okay, I think I'm going to wrap it up there. That was a great Q&A. Thank you for everybody and your really interesting questions. It's wonderful to hear about the various interests of everybody that are joining us in these Collaboration Network webinars. Thank you for Eric for hosting, as always. And thank you to Jamie. That was really informative, and I love hearing about all this great work that's being done across the state. So that's it. There will be a recording of this webinar available on the Collaboration Network's website, and you'll see it also on YouTube, I believe, right, Eric?
Yes, I went ahead and
think
put
that's
the
good.
link to the playlist in the chat. So if you missed any of the prior webinars, you can find them listed there. Thank you, Susie. Thank you, Jamie. I hope this webinar grows the interest and participation in the California Molecular Methods Workgroup. And I know that the information is going to help people with their daily chores as well, and making our waters more swimmable and more accessible to folks. Thank you so much for this. We're in the middle of Rivers Month, if you didn't know that. So this is a great thing to share with your colleagues. And next month in July, it's Lakes Awareness and Lakes Appreciation Month. So again, great topic, very timely. Thank you so much, Jamie. Thank you, Susie.
Great. Thanks, everybody.
Bye, everybody.
Bye.